Why do we use SPRM to study, design for studying cells? So because membrane proteins are important and for medical research. As a fact, about 1/3 of human proteins are membrane proteins, and they account for like over 60% of the drug targets for today's medicine. And on the other hand, SPR is actually a very well-known tool for study the binding kinetics between the biomolecules. And however, the conventional SPR requires the ligands to be fixed and uniformly fixed onto a sensor chip. So the proteins has to be purified and fixed onto the sensor chip. And most of the membrane proteins doesn't like that process. And SPRM actually provides a unique way, especially to do that, to study the proteins with proteins in their cells. And so the key challenge for studying membrane proteins is how to maintain their native environments and to keep their activity during the study. And SPRM, I will show you, and how SPRM can be used to do that. So there's 3 key components in the surface plasmon resonance microscope, SPRM. So the first one is the SPR unit with all the light sources and the detectors, and which allow you to do the SPR measurement. On top of this is a bright field optical microscope, which is looking at the same area as the SPR unit. And this optical microscope allows you to, first of all, position and locate the detecting area for the SPR. And also, it enables you to monitor the stage and the status of the cell during the experiment. The last component, which is also very important, is a uniquely designed flow system. And this flow system not only allows you to culture the cells directly onto the chip surface, but also provide precisely controlled flow dynamics, which will ensure you to have accurate. So you can see from this, SPRM actually is doing SPR measurement on a heterogeneous cells chip surface with cells sitting directly on top of that. And how that's possible. So on a conventional SPR, you may already know, so it uses a laser beam and shine a laser beam onto a metal insulator interface. And at a particular angle, we call the SPR critical angle, and then the laser beam will start to resonate with the plasma waves trapped at the metal and insulator interface. And then all the energy, all the light will be absorbed. And this angle is called the SPR angle or the SPR response signal. It is very sensitive to the reflective index of the interface and very sensitive to the changes, for example, like absorptions or reactions occurring at the interface. So that's why SPR is a powerful tool used to look at the study the binding kinetic between biomolecules. For SPRM, so instead of using a focused laser beam, we actually use a collimated light source to illuminate a fairly large area of the interface. And the SPR response from every point of that illuminated area will be recorded as a SPR image. So this SPR image actually measures the lateral variation of the reflective index across the whole illuminated area. For example, if you have a cell sitting on top of the chip surface and it has a different reflective index comparing to the neighboring area, and it will show up on the SPR image. And when you have molecules start to interact or binding to the cell surface, and it will cause the reflective index corresponding to the cell to change, and also correspondingly to see a change will be recorded on the SPRM image. And this SPR image can be recorded for the whole process of the experiment. And in this way, you can see that the SPRM actually mirrors the changes of the SPR response according to the lateral position and also with time. So once the experiment is finished, you can extract the SPR responses from any point on the image and then plot it back as a function of time. And then you will obtain the so-called SPR sensogram. And this is with this data, you can do the analysis and then derive the binding parameters from it, such as the affinity and the K on and the K off.